Um, so welcome to the March webinar of PAASTA. So this is the Paleoproteomics and Archaeology Society for Techniques and Advances. A quick reminder that you can keep up to date with the PAASTA community through our Slack channel, through the mailing list, through our website and on Blue Sky, and you can sign up for the mailing list on our website. We want to encourage a safe and positive space for the sharing of knowledge, so that we ask everyone to abide by our code of conduct, which you can find on our website. And we want these talks to really inspire connection and communication and discussion. So please only share and disseminate the results and the discussions that we have with the express permission of the speaker. And please contact the speaker directly if you are seeking interviews or statements for reporting. But this week we are very happy to be welcoming Ioannis Patramanis from the Globe Institute at the University of Copenhagen. And he's going to be presenting his PhD work on PaleoProPhyler, which is an open source reproducible pipeline for phylogenetic inference using ancient proteins. I'm going to post a link in the chat to his paper, and I'm going to hand over to Ioannis now for him to start his presentation.
Thank you very much, Helen. Just give me a second to share the screen. Hide this. Yes. Okay, perfect. Can you see the screen?
Yeah, perfect.
Thank you. So hello, everyone, and welcome to my presentation. As Helen said, my name is Ioannis, and I am a postdoctoral researcher in the Enrico Capellini group, and we are located in the Globe Institute in the University of Copenhagen, which is in Denmark, of course. Now, my main research interests are in phylogenetics and population genetics, and especially the application of those two for the study of human history. However, for the last 4 years or so, I've been working in paleoproteomics and trying to combine paleoproteomics with these three interests of mine. And you may be wondering, why would you want to do that? Why would you want to combine paleoproteomics with, let's say, phylogenetics? And my answer to this is a single word, and that is time. You see, ancient biomolecules have allowed us to study extinct species and populations, observed past admixture events, and detect genetic turnovers, which wouldn't have been possible with only using contemporary molecular data. Ancient DNA has been the main biomolecule for studying these events into the past, because it can provide us with the maximal genetic information from a specific sample or a specific population. Over the last decade or so, the field of ancient DNA has seen an unprecedented growth, owed to advances in DNA sequencing technologies and extraction methods, leading to a massive increase in the availability of samples from different times and different locations. However, like any other field, the field of ancient DNA is not without its own limitations, and on this slide I'd like to focus on the limitation of time. You see, the oldest ancient DNA data to this day comes from sedimentary data from the north of Greenland and is dated to around 2 million years ago. The oldest ancient DNA from an actual fossil, though, comes from the north of Siberia and is dated to around 1.2 million years ago. And the oldest ancient DNA from a hominin comes from the north of Spain, from the site of Atapuerca, and is dated to around 450,000 years ago. However, most ancient DNA is much younger than that and is concentrated here on the very right of the plot. Additionally, most ancient DNA comes from cold and dry regions of the world, because those are known to be more favorable to DNA preservation. Now, to address these spatial and temporal limitations of ancient DNA, a new field is now emerging, and this is the field of paleoproteomics. This field aims to study ancient proteins by extracting, isolating, and sequencing said proteins from ancient remains, and then using those sequences for evolutionary inferences. Like ancient DNA, ancient protein structure is slowly degrading, leading to missing data and chemical modifications. However, specific proteins have been shown to be far more resilient to degradation through time and because of environmental conditions. And we can visualize this difference by looking at our previous plot and then overlaying it with some of the oldest proteins that have ever been recovered. And you can see, for example, that some of the oldest proteins ever recovered actually don't even fit into this plot, because they are dated to, this plot goes up to 2 million years ago, and these are dated to 3, almost 4, and recently in a preprint pushing it up to 20 million years into the past. And again, if we look at the oldest ancient DNA samples on a map, we can see for samples that are older than 1 million years, we can see that all of them come from cold regions close to the Arctic or the Antarctic. However, some of the oldest proteins that we can recover, so again, older than 1 million years, actually come from very warm places of the world, such as Africa and Southeast Asia. And this is the promise of paleoproteomics, the ability to get molecular data from organisms that lived more than 1 million years ago and from regions of the world that are traditionally unfavorable to biomolecular preservation. And this is especially important if you're someone like me who is interested in human evolution, because most of our ancestors lived in warm places like Africa and Southeast Asia and lived millions of years into the past. However, there are a number of reasons why we should be skeptical of phylogenetic applications and results using paleoproteomic data. For example, the transcriptome, which translates into the proteome, which are the full proteins of an organism, is a mere fraction of the total genome. In humans, it's around 2%. Even then, we almost never get the full proteome. We usually get a handful of proteins, usually from teeth or bone. And then the sequences we do get are very fragmented with a lot of missing data. All of this can lead to a very limited view of the whole genetic history of a species or a group or a specific sample. And you may say, so what? It's better to have a limited view than no view at all into an organism. And this may be correct. However, I want to give you the example of only looking at mitochondrial DNA data from archaic and modern humans. If you plot a phylogenetic tree of that data, you can see that the clade of the Neanderthals is clustering closely to the clade of modern humans than to, let's say, Denisovans or the hominids from Cima de los Huesos. This may lead you to the wrong conclusion that Neanderthals were actually genetically closer to modern humans than, let's say, to Denisovans. Additionally, if you look at these two well-separating clades between Neanderthals and modern humans, you may think, or you may come to the wrong conclusion, that Neanderthals and humans never interbred with one another. In fact, this was the conclusions that some of the early ancient DNA works came to, because they only had access to mitochondrial DNA data, and now we know this is wrong based on our whole genomes that we have from these archaic populations. Additionally, proteins are under constant selection, meaning that their sequences are very, very conserved, like the ones we see here. Most mutations that happen on a gene are either silent or synonymous, so even though they cause a change on the DNA level, they do not cause a change on the protein level. These two facts together mean that there's very, very few sites for us to work with when we're doing our phylogenetic inferences and our species identification. And those sites will look something like these ones right here and these ones right here. So, with all of this in mind, let's go back to 2020, which was the start of my PhD. And at the time, there were multiple phylogenetic applications or investigations using paleoproteomic data, including hominids, some camels, sloths, ungulates, and others. However, the tools and the data necessary to evaluate the efficacy of those phylogenetic studies, and in general phylogenetics using ancient proteins, were not available to us at the time. And so we thought, so that was initially the inspiration for the project that we'll be talking about today, which is titled PaleoProPhyler, a reproducible pipeline for phylogenetic inference using ancient proteins. So as Helen mentioned, this was the first chapter of my PhD thesis, and it is now published in the peer community journal, which I encourage you to look into. And the motivations for this initially were tackling two key issues as we saw them in the phylogenetic analysis of paleoproteomic data. One, reproducibility, and two, availability. With the term reproducibility, we are talking about the fact that paleoproteomics is an emerging field, thus there are little to no standards for the phylogenetic analysis of that data. These analyses are also reliant on in-house scripts and reappropriated software, making them a bit difficult to reproduce. By the term availability, we're talking about the fact that these analyses are not really approachable to newer labs who are entering the field, and to people from very different backgrounds, like people coming from lab work, mass spectrometry, or fieldwork. Finally, we noticed there was a severe scarcity of proteomic data. Even for the people who know their bioinformatics and know their phylogenetics, they want to do it on protein data. And we can visualize this scarcity of proteomic data by looking at two online databases with different species in them. On the left, we have NCBI genomes, which contains more than 11,000 species with their full genome. And on the right, we have UniProt proteomes, which contains less than 1,000 species with a described proteome. So over 10 times difference between those genomes and proteomes. Another way we can view this scarcity of proteomic data is the availability of within-species diversity. And I like to showcase that by showing a print screen of a search I did, searching for a specific species, Pongo, and a specific protein, AMLY. And you can see here that the result was a single protein sequence that came from this search from searching all of NCBI. That means if you're someone like me who was interested in variation for that protein and for that species, you will not be able to find such online. Now, to address these issues of reproducibility and availability, we look back to some of the most recent publications of our group, including the Gigantopithecus blacki, the Stephanorhinos, and the Homo antecessor. And all of these studies try to do similar things in assembling a reference protein dataset from genomic data, aligning ancient proteins onto that reference dataset, and then finally generate various phylogenetic trees. And the code to do all of this was written by Jasmin Ramos Madrigal. So we took the code from Jasmin in the form of custom scripts and software commands. We wrote new code when possible, updated code, and then combined it all together to create a number of workflows. Our goal for these workflows was for them to be as accessible as possible. So we made everything available online through GitHub. And the user only requires to have Linux installed with Conda, a Linux machine with Conda installed. And we also wanted them to be distributable. So we packed everything into Conda environments, which contain the necessary software you need to run those workflows. We also wanted them to be automated and reproducible. So we made everything into SnakeMake scripts, which take a specific input and generate a specific output. Finally, our target audience for this would be non-bioinformaticians, but of course we wanted people with bioinformatic experience to be able to tweak them if they want and use them for their own reasons. And the result is this pipeline composed of three distinct but interlocking modules. Module number one on the very left can fetch the user reference protein sequences from a curated database, such as Ensemble, which I think a lot of you will know, as well as some additional resources, which then the user can use with module number two. Module number two can translate the proteins of the user's interest from whole genome data files, such as VCF files or CRAM or BAM files. And then finally, module number three is a fully automated phylogenetic analysis in which the user can put in their ancient protein sequences, combine them with translated proteins, and then the proteins from a curated database such as Ensemble. And then it will perform the alignment, report on phylogenetically informative sites, and then finally generate various phylogenetic trees. And this was a rough overview of what the pipeline does, but you're probably still wondering, okay, how does this work? What does this actually do? And I think the best way to demonstrate what the pipeline actually does is to try to reproduce a phylogenetic analysis of a real sample. And this sample could be Homo antecessor, which belonged to a human population that lived around 1 million years ago from the site of Atapuerca in northern Spain. And previous morphological analyses have shown that Homo antecessor is an outgroup to modern humans, Neanderthals, and Denisovans, morphologically speaking. And finally, in 2020, a study led by Frido Welker managed to recover around 1,000 amino acids spanning over 7 different proteins for one of these individuals from this species. So for now, let's imagine we were given these 7 proteins, those 1,000 amino acids, and we were asked to perform a phylogenetic analysis for that species and for Homo antecessor. What will we need for this? Well, the first thing we would need is a reference dataset. And we can get that using module number one of the pipeline. Module number one will need as an input a list of proteins, so these 7 proteins that we're interested in, and a list of species that we want to have in our reference dataset. So things that we expect to be related with Homo antecessor, like let's say modern humans, maybe chimpanzees, maybe gorillas, things that are close. And then module number one will run and fetch you from Ensemble the protein sequence from each of those species and each of those proteins. It will also fetch back some additional resources, such as the chromosome location of the gene that codes each of these proteins, and then exon and intron information on that gene. Those will be useful for us in a later step. For now, we have what we need to generate what I call the skeleton of a phylogenetic tree. And if we plot that, it can look something like this. And over here, we can see that the Homo antecessor is indeed clustering very closely with Homo sapiens, and that the other species are as related to us as we expect them to be from genetic data. However, you may be wondering if this is enough, and people might be asking you if this tree is satisfying enough, or if you can do something more. And I think most people would be asking for you to include in your tree some within species variation. So let's say more modern humans, maybe some subspecies for the other species, or maybe extinct species or populations, such as the Neanderthals and the Denisovans. But as I mentioned, there is a severe scarcity when looking for proteomic data online. So how do we get those sequences? Well, some people that are a bit smarter than me have already figured this out, and the fact that you can translate the proteins of your interest from whole genome data files. And for this, we can use the 1000 Genomes Project, which has a lot of modern humans, the Great Apes Genomes Project, which has other African great apes, and then high coverage archaic genomes from the extinct populations of Neanderthals and Denisovans. And we can download that data in the form of BAM files or VCF files, and then use them as input for module number 2. Along with the list of proteins that we want to translate and the list of samples from these files that we want to translate. Module number 2 will fetch from module number 1 the protein reference sequences for each of those 7 proteins, the chromosome location, and then the exon and intro information. And we'll use all that to finally generate the proteins of your interest from the samples of your interest from those data files. And I included this slide for anyone who is interested on what is happening under the hood in the tool. How does the translation process works? We can go it step by step as an overview. You can imagine that we start with a BAM file or a CRAM file, which these two files are quite similar in the information they contain. Or we can start from a VCF file along with a reference genome, because usually the VCF files only contain information on the variant sites. So we need the reference genome to fill in the gaps for the rest of the sites, so the tool can know what is in there, what the sequence is like there. And then module number 2 will use the information on the chromosome location, the strand, and the gene is located on. And then it will isolate the gene that codes those proteins and transform it into a FASTA sequence, DNA FASTA sequence. The next step for module number 2 is to use the exon and intro information and take this FASTA sequence, splice it, remove the introns from the sequence, and then stitch together the rest, which are the exons. And then finally, once we have the exon sequence in the form of a FASTA file, FASTA sequence, it will use a reverse blast of the DNA sequence onto the reference protein that it downloaded from Ensemble. And it will find the best triplet step to translate the protein, the DNA, into the protein, and finally generate a protein FASTA sequence. So after all of this process, we have our dataset, which we want to convert into a phylogenetic tree. And that includes our ancient proteins, our reference proteins from Ensemble, and the translated proteins we translate using module number 2. And we can use all of that as input to module number 3, which will run a slightly specialized alignment, then give you some information on your alignments, such as how many informative sites and where they are located, and then finally do the concatenation of those 7 proteins into one. It will then generate an individual gene tree or protein tree for each of those 7 proteins, as well as a maximum likelihood and a Bayesian concatenated tree from the concatenation of those 7 proteins. So using the most information that you have from all of the proteins together. And if we plot the concatenated phylogenetic tree, it can look something like this, where indeed we can see that the Homo antecessor is plotting as an outgroup to a clade involving modern humans and Neanderthals, and then that the other species are all clustering together, like the chimpanzees, the gorillas, and the orangutans. So once we were finished with the pipeline itself, we also wanted to develop some additional resources for the paleoproteomic community and for the pipeline. So we created a tutorial for the pipeline, and then we also created a protein dataset for people to do hominid phylogenetics using proteins. And the tutorial of the pipeline, which is available from this link right here, you can also find it, I think, in the paper, but you can also ask me or just Google the name of the tool in GitHub. And this is a step-by-step tutorial that is available online on GitHub, as I said, and it tries to reproduce the Homo antecessor and Gigantopithecus blacki phylogenetic analysis using the ancient proteins that are previously available from those works online. We also created this hominid paleoproteomic reference dataset, and I'm quite proud of this dataset. This is a dataset of translated proteins from more than 170 whole genomes. And those genomes belong to um, individuals from the groups of Pongo, Gorilla, Pan, and Homo, including subspecies for these groups, and including the archaic hominins of Neanderthals and Denisovans for the humans. And these specifically are 3 high-coverage Neanderthals and 1 high-coverage Denisovan that were available at the time of the publication. And I know there will be new high-coverage genomes coming. And the result was that we created a dataset of translated proteins, which includes around 1,500 proteins that I found that have been previously reported to be found in either bone or teeth. And that includes modern bone and teeth proteomes. And this data set is available online on Zenobo for anyone to use. It is citable by itself. You actually don't have to cite the paper or the pipeline. You can just cite the dataset from Zenodo. So in conclusion for this project, we have developed an automated workflow that can fetch reference proteomes from well-described organisms, translate proteins from whole genome data files, and then generate various phylogenetic trees. And we have also released some additional resources for the PaleoProteomic community to use in conjunction, including a tutorial for the pipeline and a reference dataset that people can use. And some final thoughts and some final slides before I close. I mentioned earlier in the presentation that we didn't have the tools and the data to be able to evaluate the efficacy of phylogenetics using ancient proteins. And I get this question quite a lot, especially after talks, on how much can we trust phylogenetic results using paleoproteomic data. Some people ask what is the resolution of using proteins for phylogenetic inferences. And for the people who have been paying attention, here I presented an automated workflow that can handle thousands of datasets being processed in the same manner, in the same way. So a tool. And then I also presented a dataset with hundreds of sequences from multiple and from multiple populations. So data. And yes, in fact, we have been working on this question and these issues, and hopefully we'll have a preprint coming out very soon. And the focus of this work was on enamel and collagen 1 proteins, because these are the ones that have been so far reported in the oldest of samples. So samples are older than 1,000,000 years, and those were kind of the interest for us. And primarily we were interested in how many proteins do you need to accurately resolve a clade. And we focused on the clade of humans, chimpanzees, and gorillas, and on the clade of Neanderthal, Denisovans, and humans. And we know from DNA data that humans and chimpanzees should be closer than gorillas as the outgroup. And then again, Neanderthals should be closer to Denisovans, and then the humans as an outgroup to those two. So we wanted to see if we could replicate that and how many proteins we would need to do that. And we also wanted to see what happens if you take the DNA data and the protein data from the same gene and then use them to generate a phylogeny. Do you get the same topology when you look at the DNA and the protein data, or do you get a different tree? And then finally, we looked briefly on the conservation and the amount of phylogenetic information that is available in these clusters of enamel and collagen 1 proteins, and how they compare with each other, the two groups, and how they compare the proteins within each group. And in fact, while we've been working on this, there have been 2 publications recently that have come out that are also looking into this question of phylogenetics and paleoproteomics. And I wanted to give a shout out to them. So one is a fully published work right now, whereas the other one I think is still a preprint. You can correct me if someone knows if this is wrong. But this work in particular did a good job on looking again, like us, enamel proteins in a primate-wide data set, so all of the primates. And I think it did a really good job in seeing how the degradation of the proteins affects your phylogenetic tree. So they had different levels of degradation and they were trying to reproduce the same tree. Whereas this work right here, I think, focused a lot more on the presence of within species protein diversity and variability and how that may affect your phylogenetic trees, how that may affect your species identification, and also, what happens if you plot a tree with single proteins or with a concatenated data set of proteins. So with all of that in mind, I want to thank my two supervisors for the PhD, which supervised this work. One is Fernando Racimo, who was my main supervisor, and the other one is Enrico Capellini, who was my co-supervisor and is my supervisor for the postdoc for a year now. And I always try to use their best pictures for doing that. And finally, I also want to, of course, thank the PAASTA community for inviting me for this talk. And of course, I want to thank you for your patience throughout this talk and for attending. And I'll be very happy to get any questions on any of the things I talked or have a discussion on any of these topics. Yes, this is the last one. Thank you very much.
Great, Ioannis, thank you so much.
I got scared for the moment that I had lost connection. Like, oh no.
No, it was great. It was great. Thank you so much. That was really, really interesting. If anybody has any questions, they can either pop their questions in the chat or they can put their hand signal up or unmute themselves and ask directly. Lots of questions are encouraged. I have one to kick off with. So, Ioannis, was wondering, we see quite often now with the DNA data that hybrids between kind of Neanderthals and Homo sapiens, or you have the kind of Denny individual who's a hybrid between Neanderthal and Denisovan. Do you think that in terms of, I guess this is another resolution question, do you think that proteomics has the kind of potential to be able to spot any of those individuals, or do you think what kind of impact do you think that would have on your analysis?
Yeah, so thank you very much for the question. This is actually something that I've been thinking about for, I don't know, the last couple of months, and I was actually thinking of writing like a grant to like try to set it up, because I think so far we're mostly focused on like paleoproteomics on like using specific sequences that match specific species, and I think most people have not looked into, do I get different versions of the same peptide with like a different amino acid? Like we saw that we have, it's still a preprint, but we have a work where we saw that, we got 4 samples from an archaic species, and actually there was variation between them. And actually one of them, it looked like they had the signal for both variants on the same peptide, like the same peptide with a different variant. And we think that is indication of heterozygosity for that specific position. And this is what you would expect to see if you were looking at a first generation hybrid. Like if you had a Neanderthal and human, you would expect that they would express both the proteome of the Neanderthals and the proteome of modern humans, and that for any position that differs between Neanderthals and humans, you should get the same peptide, like both peptides, and theoretically in the same frequency, if we assume that they're expressed in the same um, manner. I don't know what happens in maybe some species, they express one of like they shut down one of the chromosomes and only express one of them. But I think this is something that it has been looked much, at least in ancient proteins. I know in modern proteomics they do things like, I think we talk in that paper that there's like, if you do things for like testing your, I think blood cells for specific diseases, you can do that for proteins, and you see like the peptide for the disease. If you're heterozygous, you see both. So it is something that has been worked in modern proteomics, but I think it hasn't been worked so far in ancient proteins. But I think it has opportunities. I think it will still be contained to the very, very first generations. Like you would see it in like the first generation hybrid should be 50-50. Then the generation after that it would drop to 1/4 is from something. But I think it has some potential, yes.
Cool, thank you. I've got a couple of comments. So Kirsty says, not really a question, but great talk, Ioannis, and she really likes you're making everything so shareable and usable by others.
Thank you.
That's great. And Hyan says, hi, Ioannis, nice talk. I'm quite curious about how you evaluate the accuracy of the translated protein sequences.
Yeah, so that's a really good question. So for the data set I got to work with, I was quite lucky. Well, lucky. I chose to work with things that are quite well studied, like humans, chimpanzees, gorillas, orangutans. So we had a lot of data, and those species do have at least some sequences. Like even for the pongos, the AMLY, there is at least one AMLY sequence. And I didn't have like a comprehensive way of like programmatically comparing them and seeing like, all the sequences generated were right. But I spent a lot of time just manually, visually looking at, I translated these proteins, do they align with the reported proteins that we have from the available data sets? So just manual. But yeah, I think someone could, you know, maybe should run a evaluation if these translations actually good. But generally, just looking if you know, those proteins make sense in the alignments. And feel free to post again if you have more follow-up questions on this.
Joanna, do you want to unmute yourself?
Hi. Yeah. Great talk, Yanis. Thank you. I have one question for the module 2. If you have incomplete data, so either your VCF file has like low quality flags, or you have parts of your BAM file that nothing mapped to, but they are in the regions that you want to translate. Does it then impute from the reference, or do I get this like marked as like X's in my protein sequences? How does it treat missing data?
Yes, thank you very much. So I missed a small part, but I think I got the full question because I was just lagging a bit. But I think the question is like, what happens with there is no data in your BAM file or your VCF file is like missing. Uh, can you hear me?
Yeah, I can. Yeah, I mean, I asked about the VCF file. Sometimes you can like flag low quality data, and then the genotype would be like a dot slash dot or something. So I wanted to know how it treats these cases.
Yes. So for the VCF file, when the information of like if there's like itself, the VCF is reporting that it is bad or missing, it will translate that into like in missing data on the translation. I have a cutoff for also the yeah, no, I don't have for the VCF. I don't have a coverage cutoff or something like that because a lot of VCFs don't have that information. Um, so if the VCF itself reports that it's bad or like well, not bad and missing, it will translate to missing. If the VCF does not report the position at all, it will then look into the reference and use the reference to fill in the position. So that's a bit tricky because sometimes, people upload VCF files, and maybe they didn't identify some variants like in their variant calling. So those will be treated as the reference in the reference that you provide. For the BAM files, I have a coverage cutoff that looks at the specific like, coverage for each site. And then if it's below the coverage, it gets turned into a missing point in the FASTA sequence. And then, depending on the DNA level missingness, maybe you have a site not translating on the protein later on, and will also be as a missing amino acid. Yes, so I think that's how it works.
Thank you.
Thank you. Thank you for the question.
Yeah, if any more questions, you can pop them in the chat or you can pop your hand up and unmute yourself. Hi, Sandra.
Hi, I have a question if that's okay.
Yes, of course.
So first of all, I completely agree with Kirsty. It's amazing that you made all of this so available and easily usable for other people. Like even I was able to use it, and that's something. But I was wondering how many data sets that you have is an amazing resource. Are there any plans to upkeep it when the, new high coverage genomes come out?
Yes, thank you very much for the question. So one point on the availability is like I try to do my best, but there's still a lot of times like when you run things in a different server, there's issues. I'm happy to get questions from this, and we have tried to resolve it in some cases. But still, maybe it's not the best. Like there are some methods. Like if I had to start it from the scratch, I might have done it differently given the knowledge I have now. On the question for the data set, uh, I think I might. It's a bit of a pain because you have download the data set, put in the new data, and then re-upload it. But the translation shouldn't be too difficult, given that I have the list ready from the proteins. It will be the same proteins again, so those like 1,500 proteins. And it's quite easy if you have it set up the pipeline. You can just drop the proteins in the list, then just give it like new genomes. So it is something that if there is interest, of course, I would update it. And yeah, I mean, most of the time I do it for myself. I continue working with proteins and hominids. So, if I do it, of course, I would also upload them. Thank you.
That's great. Thank you.
And I should say, if anyone else has requests, I might be willing to, translate some datasets. I had a personal interest because we had a side project that never took off about like elephants and mammoths and archaic elephants. So I made like a dataset of elephants. It is there. No one has ever used it. It hasn't been published in like a publication or anything, but it is there in the Zenodo. But it is also a bit fun to make your, datasets. And of course, it has benefits that people use it and, hopefully they cite you and stuff.
I think this is quite an interesting question, right? Because I think you're so right that for a while now, people have just been used to each lab having their own way of basically processing data and it not being kind of reproducible. Do you, in terms of the kind of uptake of PaleoProPhyler, it's been available for a little while now. Do you know how many people have actually been kind of using it and publishing it? Is it something that if these tools do become more widely available and there's more options, then people are going to be using them, do you think? Or do you think people will kind of stick to doing it the way that they've been doing it so far?
Yeah, I think so far it's been like the second example. I think it's more like an institute. Like there's a few citations of it, and they are mostly from people from Copenhagen, or like our collaborators somewhere else. So I think it's still like a very, closed usage. And I think I like the two last the two last projects I showed. Like Johannes is one of them, but they also like did their own translations in their own way. I'm not sure how it goes. Like I'm happy with either way. There is a question of, standardizing is quite important. But if other people want to do it in a different way, it's also like an issue of trust. You trust your own data much more than you trust like random people that are on the other side of the world publishing. So yeah, I don't know. So far, it's been more closed, I would say.
Hey, do you have any final questions or comments?
Okay, I can say that, anyone who has questions, you can feel free to send me. You can also post, like, if you're using, you can technically post it on the GitHub. There's like a issue space if you're having issues. Otherwise, I think you can Google my name and find the KU mail, Copenhagen University, or Slack PAASTA. I don't check it that often, unfortunately. Yeah, or anywhere you can find me. I'm happy to talk about things or questions or anything.
Great. Well, thank you very much, Dennis. I can say from personal experience, and yes, has been very helpful and accommodating with getting the pipeline to run for, I think, sometimes there's different servers and things have clashes. So Dennis has been very helpful with that from my point of view. So yeah, I'm sure he'll be happy to answer any questions via email. And if there are no further questions or comments, thank you all very much for coming. Thank you, Ioannis, so much for presenting. And we'll be putting the talk online on our YouTube channel as well. And we'll see you all in a month again for our next talk. Thanks.
Thank you very much.
Cheers. Bye, guys.